on what's going and try to kind of better understand, how to fix it. So I'm going to talk about, some of the things that we've done in the last decade to try to build, what we think of as kind of like an ecosystem for open and reproducible science. But I'm also going to kind of motivate, in more detail, what are some of the problems that we have tried to fix? And so, some of the things that we've done, Noah mentioned fMRI prep. fMRI prep is kind of part of a larger ecosystem of tools that we've been involved. We haven't built all of them. We've been involved in building things like BIDS, the brain imaging data structure, and Open Neuro, which is the data sharing platform for data that are organized via the BIDS framework. But I want to step back and talk about like how I think about the work that we're doing. And I really think about it as a design problem. Does anybody know what this thing is?
Paper shredder?
Paper cutter. Does anybody know why it has two green buttons? Yeah.
You have to use both hands to use it, so cut your hand off.
Exactly. To prevent you from doing the stupid thing that humans will do, which is to hold the paper in like this and press the one button and cut their fingers off. So that's like, design that's meant to like, build in the kind of behaviors that we want and to avoid the kind of behaviors we don't want. And that's what good design is all about that, right? If you read like books like Don Norman's Design of Everyday Things, right? We design things to try to afford the behaviors we want to afford and prevent the behaviors that we want to prevent. And it turns out that in science, one of the big problems is that humans are really good at fooling themselves, right? Science is about asking questions. Turns out that we're pretty good at fooling ourselves about the answers to those questions. And so, this is more than 10 years ago now, people in Nature talking about, how researchers are seeking ways to fight their own worst instincts. So that's one of the ways to think about what we're trying to do is like help people fight their worst instincts. And there's ideas from behavioral economics about what's called choice architecture. The idea is like anytime you go in to behave, there are choices that are afforded and choices that are more difficult, right? You go to the grocery store, usually it's like the junky food that's right at the checkout stand, and the fresh fruit is like way back in the back, right? Because they want you to buy this and are less interested in you buying the other stuff. And so one of the ways is people have talked about this is the concept of nudges, right? You can build situations or build tools that nudge people towards doing the thing you want them to do, but you still give them the choice, right? So, if you think about it, like you can still go buy the banana, you just have to walk a little bit more, right? But by putting the candy bar by the checkout register, you're being nudged towards buying that. So similarly, we can think about like, how do we do that in neuroscience, right? Well, we can improve incentives. Incentives are hard. We can use the power of defaults, right? People are very often going like if you have a default setting in a piece of software, people are very likely to use that default setting. I'll actually show you an example of that later. Giving people feedback, right? Telling them when they're doing something that they might not want to do. And then expecting and trying to prevent errors. Like these are all things that we can try to do to help make people more likely to do the right thing. Have any of you heard of this paper, Why Most Published Research Results Are False? Okay, if you haven't, you should go look it up. It's published, 20 years ago now by John Ioannidis, colleague of mine at Stanford. He basically made an argument why exactly what he says. Most published research results are false, especially those using kind of null hypothesis testing. And he laid out a set of kind of considerations that might make studies more or less likely to be false in a particular research area. And one of them that he pointed out was the smaller the studies in a field, the less likely the findings are to be true. And we know that neuroscience research is in general pretty underpowered. And so statistical power, right, is the ability to find an effect if it exists. So if you have, we generally target studies having like 80% power, right? Which means that if there is an effect there, you will find it 80% of the time. You'll fail to find it 20% of the time. This paper by Kate Button and colleagues looked at statistical power across a bunch of studies some human neuroimaging studies, some like rodent studies, and basically found that in general, neuroscience research was really badly powered. So what this shows is that, a ton of studies that they looked at actually had, a lot of them actually had power from 0 to 10%. That means that you could do the experiment 10 times, and even if the effect is there, you're only going to find it one out of those 10 times. The other 9 times, you're going to say no. Like if you try to submit this to an IRB, if the IRB knows what they're doing, they would basically tell you can't do this because it's futile. This is what's called futile research. It has so little chance of actually finding anything that it's not worth the risk of that you're putting the subject under, to collect the data. And a ton of research like that is badly powered. Now, this was done a while ago. I think this was published 2013. This is a more recent paper from Marcus Munifo and colleagues that showed, this was looking across not just neuroimaging, but a lot of biomedical research, psychiatric, neurological, and kind of just like regular old medicine stuff, and shows that in general, you see the same thing. Like a ton, like a third of the studies are basically so badly powered as to be pretty much futile. Now, what people might say is, well, we're controlling our type 1 error, right? We're doing our p less than 0.05, so we're only going to have a false positive 5% of the time. And that's true. But there's another number that you might care about. So the false positive rate is, when you do p less than 0.05, you're basically saying across all of the studies, there's only going to be 5% false positives. But in Ioannidis's paper and then in the Button paper, they talked about another number that's important to think about, which is called the positive predictive value. This is the likelihood that if you look at only the positive results, only the results where I said, yes, I found something, how many of those are true? And what you see is basically that the higher the statistical power, or the lower statistical power, the lower the positive predictive value. So here's a thought experiment. Let's say that you're a physicist and you have a detector that's trying to detect some kind of signal out in the universe. But unbeknownst to you, your detector is broken. And so it's basically just giving you Gaussian noise. And so, you do 100 experiments and you're going to get on average 5 of those being statistically significant, right? If your threshold is p less than 0.05. But how many of those are true findings? 0, right? None of them are true because your machine's just giving you noise. So your positive predictive, even though your false positive rate is controlled at 5%, your positive predictive value is 0, right? The fact that you found a result tells you nothing about whether there's actually really a result. And as your machine, if you fix your machine, it gets better, then the more likely it is that a positive result is a true positive. There's another phenomenon that occurs called the winner's curse, which is that when you're very low powered, when your power is low, the only effects you can find are really big effects. So if you look at the literature, you'll see that people report really big effects. And that's just because those are the effects you can find with small studies. I'll show you an example of this in the wild in just a second. Just want to point out one other phenomenon, which is that when your sample size is small, your confidence in the particular value of your statistic is weak, right? Another way that is to say, the statistical estimates that you get are really unstable. This is just simulation data from Felix Schomburg from a while ago. Basically, this is just simulating data where you vary on the x-axis here. You basically each line here is just sampling another person or another sample in that particular sample that goes up to say 250 total. And what they find is that when the sample size is small down here, the true correlation is 0.25, but with small samples you can get anything from an estimate of 0.5 down to an estimate of minus 0.2, something like that, right? And as the sample gets bigger, then things start getting closer to the true value. They call this the corridor of stability. How many of you are familiar with this paper by Merrick et al., published a few years ago? So it made a bit of a splash because it basically said what the Schomburg thing had shown with simulations, which is that when you have small samples and you compute correlations, they're highly unstable. So they basically just were showing the statistical theory kind of works. That this is using some large data sets like the ABCD data set, looking at correlations between brain and behavior and showing that when you when you have large samples, you can see that, the true correlations between any particular brain thing and any particular behavioral thing are going to be really small. Like they're never going to be bigger than about 0.1. But if you take tiny samples, you can see, so this is just a couple of individual samples looking at comparing like, cortical thickness or resting connectivity with cognitive ability, showing that, you can get anything from a positive correlation of 0.5 to a negative correlation of 0.6 something, with samples from the same data set, where the true correlation is like minus 0.08 for the full data set. But just from small samples, you can get, really, really big variability. So I'm going to show you an example of a place where basically this kind of problem drove people to waste about a decade of their research lives asking questions that we now know were pretty much all the results from this field are not reliable. And this is what were called back then candidate gene studies. So back around 2000, it became possible to relatively cheaply quantify, an individual's genetic variants at particular places in the genome. And so people started doing studies where they would say, I'm interested in the hippocampus, for example, and I want to know, does the volume of the hippocampus vary in relation to this particular variant in a gene called BDNF, the brain-derived neurotrophic factor, which is known to be kind of expressed in the hippocampus, and this particular version of the gene, basically if you have one version of the gene, you're going to have more effective BDNF than the other. And so people started looking at, does the size of the hippocampus relate to whether you have one of the other versions of this BDNF gene? And there were some early studies published. The size of, sorry, I don't have a legend here. The size of the circle is basically related to the sample size. So these are sample sizes of like 20. These are sample sizes of hundreds. So this is showing over time, early on, there were very large effect sizes. Like this is a Cohen's d of 1. That's 0.8 is usually where we would say if it's above that, that's a large effect. 0.1 is like one standard deviation difference between the groups. That's a huge effect. So early on, you saw very small samples, very large effects, no negative effects being shown down here, right? Over time, sample sizes got bigger and the effect got smaller. This is actually something that's so widespread that there's a name for it. It's called the decline effect, that in general, over time, effect sizes in almost every field get smaller. And usually that's because sample sizes get bigger, people start to realize what the problems are. And probably because, almost certainly other people were computing the same correlations, and they were getting results that were actually down here in the negative side, but those weren't publishable. In fact, I remember, I was like early 2000s, I was going, I was a junior faculty, I was going around giving talks, and I had seen these papers about, there was another gene that people were very interested in, it's called COMT, related to prefrontal cortex. And I would ask people, what have you been up to? And they would say, oh yeah, we did some stuff with COMT, but we didn't find anything. And they didn't go publish the fact they didn't find anything. They just kind of set it aside. And so the only people who were finding it were the people finding these big effects in underpowered studies until others came along. We now know that none of these effects actually replicate. This is specifically for hippocampal volume, but this is true for almost every other, with maybe one exception, almost every other candidate gene association that was studied fails to replicate when you start having very large sample sizes. People in genetics in the 2000s realized that this was a problem. So now you can't really publish a genetic association study without tens of thousands of subjects, often hundreds of thousands. And that's why you have all this work being done by consortiums, and every paper has like 100 co-authors on it, because no one lab is going to go collect 200,000 subjects worth of data. But these large studies have given us the ability to actually find genes that are associated with phenotypes, but also know that none of these candidate genes actually held up. Okay, so what about fMRI? We did I have a paper back in 2017 that kind of laid out some of the issues with statistical power in fMRI. And we did some initial analyses looking at what is the statistical power of fMRI studies. And it's kind of statistical power is very hard to quantify with fMRI. And I think in general, like we've kind of come to think that power analysis for fMRI is kind of not really a sensible thing to even try. But nonetheless, you can, under some assumptions, try to at least estimate how powered was the study? So you can get sample sizes. We get these from kind of literature mining. And in 2025, the median sample size is 38 subjects per group. And notice this is actually log sample size. So it's actually been going up faster than this makes it look like. But nonetheless, when we did the paper, it was about 26. Now it's like 38. And so under some assumptions, let's say that you're looking for a single activation with 200 voxels. You have a power to find something with an effect size of about 0.65. That's a fairly large, that's kind of a medium to large effect. Now, when the question that you always have to ask is like, if you have power to find an effect, is that a plausible effect? So let's say I tell you I'm going to do a study that has power to find an effect size of, a difference between two groups with an effect size of 4. That's like the difference in height between like a 5-year-old and a 16-year-old, right? You're probably not even like, that's not a study even worth doing, right? Because I don't need statistics to tell me that 5-year-olds are shorter than 16-year-olds. That's not a plausible, interesting scientific effect. In general, plausible, interesting scientific effects are much smaller than that. And so the question is an effect size of 0.65 standard deviations actually a plausible effect? And you can't just look at the literature to tell you whether that's a plausible effect, because for reasons I'm going to talk about, because the effect sizes that are in the literature are those that survived thresholding of fMRI data. And so they suffer from that winner's curse that I mentioned. So what you really have to do is go find an unbiased way to ask what the effect sizes are. So we did that with the data from the Human Connectome project. This is, about 1,800 something subjects who we analyzed here. And it has a bunch of tasks, and we took a number of the tasks. This is just an example from the motor task. So you can take all the subjects and get for each subject, you get an estimate of the activation. And then you do statistics across subjects to ask, what's the effect size across the population in terms of like, how much is each voxel activated compared to the baseline? And the question is, how do you find which voxels to look in? Now, fortunately, the Human Connectome Project had published a paper beforehand saying, for each of these tasks, these are the regions of interest that we propose will be activated by the task. And then we also used this tool called Neuroscience. Many of you probably heard of Neuroscience. It's a website where you can do sort of online meta-analysis. So we basically went and said, we look up motor and say, where are the voxels in the brain where we're pretty sure a motor task should activate the brain? So we basically combined these two to find voxels where we're really sure that they're going to be activated. And then we can use that to say, now, how big is the difference between task and control? What's the effect size? Now, there's two different ways to kind of estimate how big an effect is with task fMRI. One is just, how much, so, when somebody starts doing a motor task, activity in the motor cortex is going to go up and then it'll go back down when they stop. And you can ask, how many like percent does it go up? We call this percent signal change. Now, in fMRI, about the biggest average percent signal change you're going to expect anywhere in the brain is like 4 or 5%. And that's going to be usually in like primary sensory or primary motor cortices. And you can see here that we see some areas that, like in the supplementary motor cortex, when people do the motor task, you get about a 4% signal change. That's a whopping effect in fMRI within a person. And that's for most like interesting cognitive studies, you would expect an effect of maybe an order of magnitude smaller, maybe 0.5% signal change at best, often more like 0.1 or 0.2. But what we really care about for our statistics is that the size of that effect with respect to the variability across the population. That's what our statistics are measuring. They're saying on average, how much difference is there between task and control in terms of standard deviations. And so when we turn those numbers into Cohen's d, which is basically a standard deviation, we see that 4%, whopping 4% signal change gives you a Cohen's d of about 0.7 something. And these others are in the kind of 0.4, maybe 0.6 range. So what this tells you is that these are things like, moving your hand versus doing nothing, or doing a working memory task versus nothing, or seeing emotional faces versus nothing. So these are kind of really whopping comparisons. And the median study published last year was only powered to find these things, right? It had a power of about 0.68. These are effect sizes in the 0.5 to 0.7 range. So what this is saying is like most studies are still, even 10 years later, 9 years later after that paper, are still underpowered to find even the most whopping effects we would expect to find. And if we look back at that Mueller et al. Meta-analysis, I'm colorblind. I should know better than doing this because it's really hard to see the difference between the dots. But I've colored the dots in green, the ones that have at least 20 observations per group. These are comparing, healthy and depressed people. The red ones are the ones where one or the other or both of the groups have fewer than 20 subjects in them. And I have this next to a quote from a paper by Simmons and colleagues. This is a famous paper published back in 2011 called False Positive Psychology, which is about a lot of the same types of issues that were happening in the psychological literature. And they basically point out that samples smaller than 20% are simply not powerful enough to detect most effects. And so you would never decide in advance to go just collect 20 subjects. And so what's almost certainly happening here is people are doing this thing that they called interim data analysis and flexible termination, where they're doing their statistics after every subject, and when they get an effect, they stop. And we know from their paper, they did simulations showing that this can greatly inflate your type 1 error. And so, we think that at least part of the reason why there's just why so many of the results in this particular meta-analysis and in the earlier literature in general are unreliable is that, that the sample sizes were too small. Now, I should say that this is particularly a problem for correlations and for group differences. It's less a problem, if you just want to know, what brain area activates on average for some particular task manipulation. Often, so for example, I'm going to show you faces versus houses, right? I don't need a lot of subjects to see that because that's a pretty whopping effect. It's really when effect sizes get smaller or when you start trying to pull apart groups or pull apart correlations where these power issues get really bad. We also know that this is a problem in machine learning analyses. Gael Veracqua had a nice paper a few years ago that showed just how variable the results are from sample to sample in predictive accuracy measures using fMRI or MEG, and that it kind of fits with what you would expect from simulations, as he showed. But the idea is that similarly here, you can get a huge, variability just from sample to sample in accuracy when you apply a machine learning tool. And he showed that you see the same type of decline effect in papers that look at classification accuracy for various types of disorders, psychiatric disorders, Alzheimer's. And you can see in general, as sample sizes get bigger, the reported accuracies go down. And we think that's in part just because when you have small sample sizes, you can get a lot of variability. Combine that with publication bias, where they either do a bunch of analyses and only report the one that's best, or they, people who find bad results don't publish them. People who find good results publish them. And so it's just basically all this adds up to say it's really difficult to trust results with small sample sizes, at least group statistical analyses. So, I wrote about this, and when many of you have probably had this experience where somebody comes and gives a talk at your university, they're like colloquium speaker, and then the trainees go to lunch with them, right? And so I would get up and give a talk about this stuff, and then I'd go to have lunch with trainees, and they're like, well, my PI only has money for me to collect 20 subjects for my dissertation. What am I supposed to do? You just told me I can't do that. And that's like a difficult conversation to have, right? So I actually wrote this paper published back in 2019 called The Cost of Reproducibility that tried to address that issue that would come up in these discussions. And I don't think the answer is to go do underpowered science. The answer is to figure out what you can do that will give you kind of reliable answers. And I love this quote from Nancy Canwisher. It's kind of a takeoff on the Crosby, Stills, Nash and Young. I think, if you can't answer the question you love the question you can answer. And so there's, some of the pivots are collaborating, right? Finding other people who have relevant data who you can kind of combine data with, like people in the genetics literature have done. Using shared data, I'll mention some of the shared data resources that now exist. Focusing on theory and computational modeling. And I think one other that I forgot to add here is like, zooming in and doing like much more detailed studies of a small number of individuals, as opposed to trying to look at large groups. And we're seeing now that that's an approach that started to pay off, what we call, dense phenotyping or intensive imaging. Yeah.
Question.
Yeah.
So dense phenotyping would still require to collect a lot of trials, a lot of hours of them. MRI scan. That again,
Yeah,
could be like it's a standard thing, right? So
yeah, it's not. I mean, yeah, there's no free lunch. That's exactly right. I mean, but I think still you can, like, if you have data to collect 100 hours of imaging, you might be better off collecting 10 hours off of 10 subjects than 1 hour off of 100 subjects. It really depends on what you're trying to get at, right? If you really care about individual differences, then dense phenotyping is not going to help you. If you really care about individual differences, you're really going to need hundreds, if not thousands, of subjects. And so unless there are shared data resources that can answer the question, you're going to be in trouble. But no, that's a good point. Yeah, it's not a magic bullet, but it can. I mean, I think a lot of groups have moved. Like my group has moved completely now towards dense phenotyping for our kind of like cognitive imaging studies. In part because we want to be able to see answers at an individual level and then quantify how prevalent is that effect across people, as opposed to just averaging and saying what's the group effect. Any other questions? Yeah.
With very large sample size and tiny little difference will be significant, right? Is that also a problem?
Yeah, this is an interesting point. So so it's not just machine learning in general. If you're doing statistics, the bigger the sample size, the higher your power, and that means it's easier to find small effects. So this is, people have Paul Meehl talked about this in the 1960s, like this idea that basically as your sample size becomes bigger, you can start to find significant effects that are like so small that they might be meaningless. And this is actually something that's been pointed out in the context of the ABCD study, which is this study that has, usable data from like almost 10,000 kids, behavioral imaging data. And the ABCD people, the group that does the analytics for them, wrote a paper a few years ago, kind of laying out this exact point, right? That you can get highly significant p less than 0.0000 whatever, when the effect is so small that it's kind of like useless. Like there's similar things that happened in the literature on social media usage, right? Where you can see like significant differences in some kind of psychological function between kids who use a lot of social media and kids who don't. But it's on the order of like, like eating a bag of potato chips once per year or something like that, like a very tiny effect. So yeah, that's a very good point. And so one of the things that you have to think about is, there are different types of statistical tools that one can start using to quantify not just the p-value, but to quantify kind of the evidence level. So like using Bayesian statistics. So that's one of the pivots that people often try to do. But it's also the case that just because the effect size is tiny doesn't mean it's not important, right? Like, if you can have a 0.05 standard deviation effect on kids' educational outcomes, right, at scale, that could be massive, right? So I think we shouldn't just discount an effect size because it's tiny. It really depends on what the impact of it might be. Other questions?
So would you say that there's like a magic number, like for a number of samples, like around 200 at least, for like a effect size?
I don't want to say that. It really depends. Ultimately it depends on the size of the effect that you're looking for, right? And so, and that's going to, for like brain behavior associations or gene behavior associations, those effects are usually really small, right? Which means you need thousands probably, right? Occasionally, depending on what you're studying, if it's a difference between two groups that are very different, people with like full-on Alzheimer's disease versus healthy aged people, the sample sizes are not going to need to be as big. So it really depends on how big the effect is you're looking for. So I don't want to give a number. Other questions? Yeah.
what kind of features that I'm using. Like it varies for fMRI, or in case if I use a structural connector from a diffusion model, or if I go for.
So the question is, right. Does it matter what type of data you're using? It matters in one way, I think, right? And that's how big are the effects, right? It is almost certainly the case that in structural MRI, the effects can often be bigger sometimes because structural MRI data are much less noisy than functional MRI data. But not always. Like in the Marek paper, they showed that correlations between structure and cognition, or between resting fMRI and cognition, were kind of on the same order. So it really depends on the exact thing you're looking at and how big the effect is. If the effect sizes are the same, then the statistical outcomes are going to be the same. Okay, so one of the pivots I mentioned, right, was using shared data. And fortunately, like the amount of shared data available now is pretty amazing compared to what it was, even a few years ago. Things like Human Connectome Project, at ABCD, the UK Biobank, Open Neuro that we run, the Natural Scenes data set that Kendrick Kay, who you're going to meet later this week, developed. Lots of resources. Obviously, there's not going to be data to answer any question you might want to answer, but often there's at least data to get started with, and then use that to kind of get your feet wet and like really learn the methods, and then go sort of apply those methods in a sample that you might collect. Okay, let's go back to Ioannidis. Another thing he mentioned was flexibility. The greater the flexibility in designs, definitions, outcomes, and analytical modes in a field, the less likely the findings are to be true. This is a table that was in our 2017 paper. This is basically just looking at the amount of analytic flexibility in fMRI, right? So this was looking just at the FSL package, at all the different, kind of steps where you have choices to make, and the number of plausible options at each of those steps. Right. None of the choices here are choices that anybody would immediately say that's a really bad choice. At least most of them, that's true. Multiply all these out and you have almost 70,000 different possible workflows. And let's say even if half of those are implausible, that's still more than 30,000 workflows. And we know from work that Josh Karp did a while ago that people actually take advantage of this flexibility. He looked across 241 papers at how often people did different things, right? So he looked at like which package they use, what smoothing kernel they use, what inference p-value they use, and he saw that there were a lot of different choices that were being made, and there were nearly as many unique analysis pipelines as there were studies in a sample. Almost every study used a different set of analysis procedures. And he showed in another paper that this actually has an impact. So here he took one data set, one that we had shared earlier, and he ran it through almost 7,000 different workflows. And this is the range. On the left side, you see the average activation map across those this is for a response inhibition task. It's like comparing stop and go trials in a stop signal task. And that looks like a reasonable activation map for that task based on what we had done before. But if you look on the right, you see basically how big are the differences from the highest to the lowest statistical value across those 7,000 workflows. You can see in some areas, they're massive. This is like, 7, 8 standard deviations of difference in the result from the highest to the lowest. You see it, and it's not across the brain consistent, right? It really varies. Much of the brain, there's places where there's not much activation, you don't really see much difference, but places where there's activation, you can see huge differences. So it actually matters what you choose. And it isn't just fMRI, right? People have talked about this in Luck and Gasplin talked about this in the ERP literature, how to get statistically significant effects in any ERP experiment. Now, one of the things you can try to do to fix this is to pre-register your studies. How many of you know about pre-registration? How many of you have done a pre-registration? How many of you have done a pre-registration for an imaging study? Wow, okay, cool. So it is really uncommon to see pre-registrations for imaging studies. Now, I'm glad to see so many people have done pre-registrations in general. And I think they are becoming, a much more kind of standard mode of operation. So the idea is, if you're not familiar with pre-registration, it's really just like writing down what you're going to do for the study and then putting it in a place where it can't be changed without someone noticing. Something like the Open Science Framework, which is where we usually pre-register things. And preferably you say how you're going to analyze the data. Even better, you actually have the code that you're going to use to analyze the data that you've developed based on like simulated data. Now, one of the concerns people often have is, well, then I can't go look at my data, right? I can't go explore the data. I can't learn from it. And that's just not true, right? What pre-registration does is it holds your feet to the fire about what you say you planned versus what you actually did. You can go explore the data all you want. You just have to describe those analyses in the paper as exploratory analyses. You can change what like if you decide something is pre-registered and you decide that's really not the right way to analyze these data, you should analyze the data in the right way and then say in the paper, we deviated from the pre-registration in this way for this reason. We've done that in pretty much every paper we've ever pre-registered. And, I shouldn't say it's never a problem because I have seen places where reviewers get upset about it. For the most part, it's never been a problem for us. This is a pretty impressive result showing the effect of pre-registration. So back in 2000, the National Heart, Lung, and Blood Association, the NIH institute that funds like cardiovascular research, started requiring clinical trials to be pre-registered. Before then, people could just go do the trial and then report it however they want to report it. Starting in 2000, they had to go to this site called clinicaltrials.gov and register, basically say, what's the primary outcome you're going to be looking, you're giving people a drug. Are you trying to change the all-cause mortality, just how many people die? Are you trying to change some particular, disease outcome? What are you trying to change? Previous to that pre-registration requirement, the majority of studies had reported positive results. That's these guys with the plus sign. Nobody had reported a negative, like not negative null, but like a negative outcome, like the drug makes you worse than you would have been, right? And a few of them reported null. That's the gray things here. After 2000, suddenly the large majority of studies published null results. That's not because drugs stopped working in 2000, right? It's because people could no longer move the goalposts to find a way to write a paper that said that that the drug worked. Now, pre-registering imaging studies is hard, and because there's a lot of stuff, and you don't always know up front exactly what the right way to analyze the data is going to be, especially for task fMRI, because there's often like modeling choices that have to be made. So we've almost always had to deviate from the pre-registration. What we've started doing now is actually collecting, like for our data set, let's say we're going to collect 50 subjects. We collect 5 subjects as basically a discovery data set where we design all the analyses based only on those 5 subjects. And then once we have the analysis working in the way that we want, we kind of do a second registration saying, here's how we're going to analyze the rest of the data. And then we go analyze that validation data set or analyze the full data set, however we had pre-registered it. And that's one way to that in general, that kind of discovery validation sample idea is a good one. We do that regularly in behavioral studies now, which is easier. You can collect a lot of behavioral subjects online pretty easily. So we'll generally collect a pretty decent discovery data set, analyze that, figure out exactly how we want to analyze it, then pre-register that and analyze the rest. Okay, so pre-registration will prevent people from taking advantage of analytic variability, but it doesn't get rid of it, right? Some groups are going to analyze things differently than others just because they have different philosophies about how it should work. And so we wanted to know, like, if you give people data like in the real world and have them analyze it, how much of an impact do different analytic approaches have? So we did a study that published a number of years ago now. This was led by Rotem Botvinnik-Nazer and Tom Schoenberg in Israel with a bunch of behavioral economists and then some other neuroscience people, which basically aimed to ask this question. So we collected a data set on a task that we've used in our lab before. I'm not going to say the details don't matter. It's a decision making, like a gambling task. You see a gamble, you say yes or no. About 100, I think 108 subjects. And then we recruited people from social media, from conferences, from the kind of like neuroeconomics world, so people who would have some knowledge of how to analyze these types of data. And we basically said, we want teams to analyze the data. You can have up to 3 people on the team. They had to sign a non-disclosure agreement basically saying they wouldn't share their results with anybody else until the embargo was lifted, because we didn't want people kind of, biasing each other. And everybody who submitted results was offered co-authorship on the paper. That's why the paper has about 200 authors. We gave out the data to 82 teams. We got results back from about 70, I think from 70. And we basically asked them to give us yes/no answers to 9 questions, which were basically hypothesis tests like, is there significant activation for increasing the size of the gamble in the ventromedial prefrontal cortex? We didn't tell them how to do it. We basically said, ask that question like you would in a study in your own lab. And it had to be whole brain analysis. That's the only stipulation we had. We got data back from 70 teams, and we asked them to give us a bunch more stuff other than just those, beyond just those yes/no answers, like the statistical maps before and after thresholding, so we can see a little bit more about what they actually did. When we looked at the workflows, we saw none of the 70 teams use the same workflow, right? There were 70 different teams. And what was really striking is, I've been involved in kind of developing these checklists for best practice for like describing neuroimaging analysis. This is this thing we call the COBIDAS report that came with this like huge checklist. So we had the people in the study give us, sometimes 15-page long descriptions of their analysis methods following this checklist. Often we still couldn't tell exactly what they did. We'd have to like ask for reasons I'll talk about in a moment. We had to go and ask for like the files from their analysis to dig in and see exactly how the analysis was done. So this is the main result. The y-axis here is the proportion of teams saying yes across the 70 teams. If everybody agreed, these numbers would all be 0 or 1. Either everybody would say no or everybody would say yes. Clearly, none of them did they agree on everything. For 3 of the hypotheses, most teams agreed that there's nothing going on. For 1 of them, they agreed that, most like 85% of teams agreed that something was going on. For 5 out of the 9, it was 20 to 40% of teams saying yes, right? Same data, same question, but 20 to 40% of teams said yes. That seems like a problem. And we saw this at the voxel level too. If you look at each voxel and say, what proportion of teams said yes? This is for like the best one, or one of the best ones, one of the 2 best. And the best voxel had overlap of like 76% of, so there was no voxel where more than 76% of teams said there's a significant effect in this voxel. So we looked at the underlying kind of statistical maps across the whole brain, and this is a correlation map. So each cell in this big map is the correlation between 2 different teams, right? Each of these rows or columns is a team, and the green and red up here is whether the team said yes or no for this particular outcome. So what's pretty striking is like teams could have like these 2 teams right here have highly correlated underlying statistical maps, but one of them said yes and one said no. But what's even more striking is the fact that there's 8 teams here that are correlated with each other, but actually negatively correlated with the rest. That's what the blue means here. So like most teams here, 60 or so, or sorry, 50 or so, were kind of pretty correlated with one another, even though they often gave different answers. Their underlying maps were pretty correlated. These were actually negatively correlated with those. And if you look at the average maps for each of these groups, you see what's going on. Like this is the map for the groups that are kind of mostly got it right. This is the map for these groups. And when we saw that, we're like, well, that just looks like a task activation map for almost any difficult task, right? You get visual activation here and anterior insula here. You get negative activation in the ventromedial prefrontal cortex, like default mode area. So we went and basically dug into what they did, and they basically put the model together wrong. They misspecified the model, and I'm happy to say more about how they did it, but I'm not going to go into detail unless anybody has questions. But this is the ones where we had to like get their files to figure out exactly how they had built the model. Nonetheless, if you do a meta-analysis across all the data, even including the people who got it wrong, you can get kind of, reasonable results. And the meta, this is like a very early version of this kind of same data meta-analysis. Tom Nichols and Camille Mamey and others have gone on and developed better tools for this now. But this has kind of led us to this idea that we should really not be thinking about a single workflow for any data set. We should really be thinking about how consistent are results across workflows. And I'll show you examples of how we do that in a moment. Just want to say that that's kind of one of the ways that one can think about, addressing the question. Other people have thought about this in other ways. So that's the approach that we took there is what's come to be called a multi-analyst study. Those are, I wouldn't recommend going and running one of those because they're a real nightmare to coordinate a bunch of different groups. But doing what we call multiverse analysis, where your group takes a data set and runs it through a bunch of analyses, that's definitely something that we've started doing as a standard procedure, and I think is worth looking at. Other ways that people have tried to address this, so Rob McCune and Saul Perlmutter had a piece about using what's called blind analysis, which is used in physics very often, where basically you're given a data set, but you don't know exactly what's in it, or it might have been tweaked, and multiple groups are given slightly different versions of the data set, or just differently labeled versions of the same data set, and you basically see, do they get the same answers? Another way to do it is using simulated data to basically run experiments and make sure that your tools are working before you actually apply them to the real data. I'll say a little bit more about, actually, let me just stop and see if there's any questions before I go on to multiverse. Okie dokie. So multiverse analysis is just the idea of running a bunch of plausible analyses on the data set and asking how consistent are the results. And there's people use various terms for this. Some people call it sensitivity analysis, specification curve analysis. Ioannidis referred to it as a vibration analysis, looking at how much the effects vibrate across different workflows. I'll show you one example that we published a couple of years ago of doing this. So one of the really, if you want to look at individual differences, the reliability of your measures is really important, because the reliability of a measure basically caps the kind of correlations one can find with it. You can think of reliability as like, how much does a measure correlate with another version of itself, right? So I have two different measures of fluid intelligence on a person. Nothing else can correlate more strongly with fluid intelligence than another measure of fluid intelligence, right? Unless something is really wrong there. And so that's why reliability really matters. And people have focused in the literature on kind of quantifying reliability of these different data types. Structural MRI are highly reliable. That's what you see over here. These things have reliability of like approaching 1. This is task fMRI reliability. And this is from the Human Connectome Project and the Dunedin study, a paper from Max Elliott and colleagues from a number of years ago, showing that roughly it's rarely above about 0.5. So that means that your correlation is capped. Like nothing else is going to be able to correlate that fMRI data better than about 0.5. If you see that it is, then something probably went wrong. So we wanted to know, like, how does this relate to analytic choices? And in particular, Michael Dimidenko, who was a postdoc in my lab, was really interested in the monetary incentive delay task. This is a task that's been used in the kind of addiction literature to get at kind of reward responsiveness, reward anticipation. It's one of the tasks used in the ABCD study for task fMRI, one of the just 3 tasks that they choose. So we took 3 different data sets with subjects doing this task. And we wanted to know, like, how does test-retest reliability relate to the different ways that one can analyze the data? So Michael had reviewed the literature to look at all the different ways that people actually analyze the monetary incentive delay task to get at reward anticipation. And it turns out that there are a lot of different ways people do it. And so we looked across these 3 data sets at 240 different pipelines, varying how much we spatially smooth the data, how we model motion, how we specify the task model, and then what contrast, like what comparison are we looking at for the task model. I can say a little bit more about those. But basically, this is what's called a specification curve. So this just shows from lowest to highest, the reliability estimates, the test-retest estimates. You have 2 measures, 2 task measures how much do they correlate with each other, basically. And you can see that this is from worst to best. So for the worst pipeline, basically there's zero reliability. You could run as many correlations as you wanted. They're never going to find one that's real because the data aren't reliable. The best you get is about 0.45, which is roughly on par with like what had been shown in the Elliott paper. The thing at the bottom here is what we call a specification curve, which basically shows you for each little tick mark here is like showing what choices were made and how do they relate to reliability. So what this means, for example, is like you can see here for this particular contrast, which is small gain versus neutral, like a chance to win a small amount of money versus nothing. For the most part, all of those pipelines had really low reliability, right? Because this is basically saying the best one maps onto this, which had a reliability of about 0.1 something. Whereas, for the large gain versus baseline, they mostly had, relatively higher reliability. And similarly for contrast, the contrasts vary a lot, but some versions of them had either, very low or very high reliability. So what this shows is that depending on the way that you choose to analyze the data, you could end up with results that have some chance of giving you reasonable correlations, or you can end up with results that have no chance of giving you reasonable correlations with any kind of behavior or any other variable. Okay. Okay, we have 30 minutes left, so we're going to move on to the section on coding, given that you're here this week to do a lot of coding stuff. How many of you have actually written code in the context of your research? Pretty much everybody. You probably wouldn't be here if that weren't true, right? How many of you have had any training in software engineering? How many of you have ever written a test for your code? Oh, wow. Oh, good number of hands for the test. Good. Okay. So, I've become convinced in the last number of years that software is a major point of risk for us in science. Everybody is right. Everybody in this room, everybody in pretty much every building that's at least doing biosciences on campus is writing code for their research. Almost none of them have been trained how to write code well. And we know of examples where it's gone wrong. This is Jeffrey Chang is a structural biologist. I think he's still at the Salk Institute, who in the early 2000s had a set of papers in Science, PNAS, other places on the structure of this particular ABCD transporter protein. He's a structural biologist. They figure out the structure of proteins. All those papers had to be retracted because of a problem with a piece of code they were using that just changed a sign. And that sign changed the structure of the protein. Somebody else tried to replicate it, couldn't, started asking questions. They realized, oh, there's a problem with the software. So 5 papers had to be retracted in total because of this one tiny little sign change in a piece of code. And we've seen examples in my lab of this. In fact, this was one of the early ones that got me a lot more interested in thinking about problems with software. So we were doing a paper to look at our ability to classify what task a person was doing. This was using data from an earlier version of Open Neuro, where we had people doing one of 23 different cognitive tasks. And we had fMRI versus baseline for each of those. And we said basically, how well can we classify in a leave one subject out way, which tasks they're doing? And I wrote some code in Python and I got 93% accuracy. Wow. If it had been 100%, I would have known I did something wrong. 93%, I should have probably been a little bit more skeptical than I was. I was still pretty skeptical, but so Sammy Koyajo, who at that point was a grad student, I think he hadn't joined the lab yet. He was still a grad student. He took the code. He was a co-author on the paper. He took the code and poked around and said, oh, hey, you did something dumb. Mm-hmm. You left out a colon and a comma, right? So I should have had colon comma train here, but I said data train. So it was doing the indexing wrong. And when you do it right, it gives you 53% accuracy. Now, 53% on a 1 out of 23 problem is still really good. So the paper was published with the 53%, and that's an interesting finding. But we very easily could have ended up publishing a paper with a completely incorrect number. Here's another example from something that happened in our lab where we had we were doing an analysis of the ABCD study. Another one of its tasks is that they use for fMRI in the ABCD study is the stop signal task. And we wrote a paper that laid out a bunch of problems with the way that task had been implemented in the ABCD study. And we put it up on bioRxiv. We'd also submitted it to eLife. And we had basically identified this particular issue for 1,300 out of the 7,200 subjects, that there's this like gross violation of a particular assumption that one has to make to actually compute the thing one computes on a stop signal task. And as you might imagine, the ABCD people weren't particularly happy to see this preprint. And so their team started digging into the code, which we had also shared. And Sage, one of their developers, found an error. This is the actual snippet of code. It's like this horrible, like multiple Boolean. And basically one parenthesis was in the wrong place. And it ended up like basically misclassifying. And so the ultimate number was 447 out of 7,000 rather than 1,300. Still enough to be concerned about. And it was just one of the 9 problems that we had laid out. So the paper ended up getting accepted, though fortunately, this happened while the paper was under review. We were able to fix the problem, rewrite the paper, and basically send it to the journal and say, hey, we discovered this problem. Please send this updated version to the reviewer. So they were actually able to review the updated version rather than us having to go back in the second round and say, oh, sorry, we screwed this up. I'm certain that these kind of errors are happening all over the place, right? And there's a problem, which is that think about what happens if you get an error that causes you to have like 0% classification accuracy. You're probably going to go figure out what's wrong with the code. Why is my classification accuracy so bad, right? If you get an error that causes like really good results, like what you expected, you're probably a lot less likely to go dig in and figure out what's wrong with the code. So I call that, it's not the greatest term, I call it bug hacking, on analogy to like p hacking. Like p hacking is like finding the analysis that gives you the best p-value. Bug hacking is like, finding the code that gives you the best results. But the point is that bugs that confirm our predictions are much less likely to be found than bugs that disconfirm them. So we've tried to assume this stance I call the principle of assumed error. And you can imagine how my students feel about me telling them this. Like whenever you find a seemingly good result, go try to figure out what the bug was that actually gave you that result. And on more than one occasion, we have found bugs that actually gave us the result. And so it's impactful. But it prevents us from publishing things that have bugs. And I think the more code you're writing, the more likely that those bugs could be occurring. So how do we try to make ourselves write better code? I mean, in part, you're going to learn a lot of stuff this week about this, right? And I've been thinking a lot about kind of coding quality. I'll talk tomorrow about, AI-assisted coding, which is kind of like the elephant in the room when it comes to coding right now. And how we can use those tools to try to actually write code that is better, not worse. But some of the things we want out of code is like, we want it more than anything else. You want code to be readable, right? You want to be able to look at it and see what it's doing. A question or just stretching your hand?
Just the question about the module. I can maybe also we inject some test code validation. Maybe because we know what's the expectation.
Yeah. Yeah, I'll talk a lot about that tomorrow. I think testing is essential. In fact, if you go, I'm going to mention my book on my Better Code, Better Science book. And the fourth chapter is all about testing. It's actually before the chapter on using AI to write code, because I think you need to be thinking about testing before you start using AI. But yeah, testing, I think I say well tested here, but testing is like essential. Yeah. I think the other thing to realize is that, coding and, tomorrow I'll distinguish between coding and programming. I should really say programming is a craft, because increasingly, a lot of us aren't going to be like writing a lot of code by hand, right? If you're using AI tools, but there's more to like programming than just generating code. Programming is about understanding the problem, figuring out how to solve the problem. And the only way to get better at this stuff is just doing it a lot, right? It's like it's a craft. So, I think one thing that's really important to make code readable is like well-styled code. Fortunately, AI tools write in general pretty well-styled code. Um, I would suggest that, if for most of you, working with code, whether you write it or an AI tool writes it, is going to be an important part of what you do for a while. I would suggest you spend a little bit of time like learning about software engineering. And I would say like these two books on the right, both of which I think you can find like PDFs online. One is called The Art of Readable Code, which is about basically how to write code that's easy to read. And then this one is by my colleague John Osterhout called A Philosophy of Software Design. I don't actually agree with everything he says in the book, um, but but I think it's a really great book for laying out how to think about what coding means, what clean coding means, what it means to write good code. And then this is kind of, the book Clean Code is kind of like the Bible. There's a new edition that just came out that actually is much more kind of AI focused. So I would suggest finding that. It's like 1,000 pages, so it's not an overnight read, but it's really well written, and I think you'll find it helpful. So one of the things that you often see is like people saying, well, I really want to release my code, but I need to add comments to it, right? People think of commenting as like a marker for good code. And if you read the software engineering literature, that like nothing could be farther from the case. That, comments are really thought of as kind of like a Band-Aid as opposed to a marker of clean code, right? So here's a couple of quotes. This is from Bob Martin, the clean code guy. Clear and expressive code with few comments is far superior to cluttered and complex code with lots of comments. Or from Kernighan and Plagard, who wrote this Elements of Programming Style. Don't comment bad code, rewrite it. The idea is like, people should be able to know what your code is doing just by reading the code. Like comments are there really to tell the reader things that the code can't tell them on its own. Things about what's the intent? What are you trying to do here? What are things I need to know? So in my book, I have a bunch of stuff about, which I'll give you the link to in a bit. A bunch of stuff about like, when to comment, when not to comment. There's also a lot of tools that one can use to try to help make code better. Because nobody wants to go read the style guide for like how to how many spaces should I have or how many lines should I have between things. So fortunately, there are tools like Rough, like Flake8, where you can just give it your code and it will take your code and make it better looking at least. It won't actually make it function any better, but it'll make it more style compliant and make it look better. And they can also check for various errors, so they can save you from like, especially if you have long running code, you don't want to wait 10 minutes for your thing to crash, only to see that you like misspelled a variable name that the static analysis program could have caught before you ran the code. One of the things that's really important, especially for learning to code, is getting review of one's code. And so there's various ways to do code review. Increasingly you can ask an AI tool to review your code, and they're actually pretty good at it. But it's also a great way to kind of like learn together with other people in your group. So in my lab, we often will do kind of a group code review where somebody will bring a piece of code, and then I or someone else will kind of just mostly walk through it. It's not so much like finding bugs. It's really about just like, can I understand what the code is trying to do without you having to tell me? And can you answer, increasingly, it's also, can you actually tell me what this code's doing? Like, if you wrote it with AI and you can't tell me what it's doing, then I don't think you should be trusting it for your science, right? So it's really about like readability and understandability. And occasionally we might find a bug, but that's not the main goal. So there's a nice paper by Petrie and Wilson, talks about like some of the kind of ways to run a code review in science. So testing has already come up. I'll talk a lot more about testing tomorrow. There's various types of tests, and testing is like a deep rabbit hole one can go down. And I think that there's, in the book, I talk a lot about, I think the ways in which you could use it to the best effect. Really you can never test for everything, and tests tell you where the code is wrong, and they don't tell you where the code is right necessarily. So you have to write the appropriate test to find the bugs that are most likely to exist. Here's an example of where like a really simple test could have saved someone a lot of grief. This is a paper published a number of years ago in PLOS One that had to have a published correction. In PLOS One, you have to share the data. You have to publish the data with the paper. So they published the data. Somebody went and had a look at it and said, "Hey, something's wrong. Your data set is supposed to be adults, and it has somebody with age 5 and someone with age 32757. That's not good." And so in the previous paper, they had said, well, age didn't correlate with any of the variables of interest, but it turns out that when you fixed age, it does correlate with some of the variables of interest. And so that's why they had to do this correction. If they had just put something into their code as a guardrail to basically say, hey, if it's an age variable, it should be within the ranges of whatever our ages are supposed to be from the study. Let's say it was 12 to 120. And if something falls outside of that, this assertion is going to catch it, right? So anytime there are like things so for example, if you're working with counts or reaction times, right, those should never be negative, right? There's various things you can try to do. And there's actually tools now, like in Python, there's this thing called Pydantic that's really good at like validating data. I'm not going to talk about it here, or tomorrow, but it's in my book. There's a couple of sections on it. So anytime you can build in validation around like the things that should be true of a particular variable, you should do that just to protect yourself. Now, there's an idea that has become increasingly popular in the context of AI tools. It's been around since around 2000. Called test-driven development. So like most of us, I think, when we're going to write code, we go write code, right? We say, what's the problem I want to solve? How can I write some code to solve that problem? Test-driven development, and then if you're going to write tests, you might come back after the code's written and say, now I need to write some tests to make sure that it works. Test-driven development flips that on its head. It basically says, you're going to start by writing the tests, right? So the tests are going to say, does the code give me back the answer that it's supposed to give me back? Then you go write the code. And the tests are supposed to fail up front, right? So it's called they often call it red-green refactor. I don't think I have that here, but the idea is like the first thing you do is write tests that fail. Then you go write code that makes the test pass. And when that happens, you're done, unless you want to go back and like make the code cleaner, or you want to go back and add more tests to add more behaviors to the code. It turns out that this is a really good way to get AI coding agents to do what you want, and in particular, to not go do a bunch of extra stuff, right? Because the idea is like if you're only doing the things to make the test pass, that prevents you from going and doing a bunch of extra stuff without having written tests for the extra stuff. And you can kind of, if you have a specification that tells the AI agent what to do, they're generally going to take that and turn it into tests and just solve those tests. I'll say more about this tomorrow, but it's become test-driven development's a little controversial in part because like some of the more zealous advocates have basically like shame anybody who doesn't do it in exactly the way that they think it should be done. But it's turned out to be really, really useful in AI. So here's the book I keep mentioning. There's actually, I forgot, I thought I had added the new link. The new link. If you actually just go to bettercode-book.org, or just Google Better Code, Better Science, you'll find it's an openly available book online. It's basically done, except there's a chapter on coding with AI that I've written twice, and it's become outdated within a few months. And so I'm going back and rewriting it again now to try to make it a little more future-proof. So that chapter is a work in progress. Everything else is pretty much done. Sorry, question? Okay, I'm just going to finally mention kind of one other issue. So I've talked a lot about reproducibility, right? Where by that we usually mean, can you get the same answer from the same data? And there's various, this is this well-known kind of like figure. I think I stole this from Wikipedia or maybe from my other book. But basically, something can be reproducible but wrong, right? Reproducibility doesn't mean that it's giving you the right answer. So in measurement theory in psychology, we talk about reproducibility versus validity, right? Reproducibility is, does it give you the same answer every time? Validity is, does it give you the answer that maps onto the thing you're really trying to measure, right? So this is a measurement that is like, in C that is like reproducible, right? It's pretty clustered, but it's not hitting the bullseye. It's hitting somewhere off to the northeast. And so we have to think not just about can we get the same answer, but is our answer correct? And that takes a whole other set of tools. And I have a whole chapter about validation in the book that I just mentioned. But ultimately, I think one of the most powerful things you can do is learn how to do these what we call parameter recovery studies, where let's say you have an analysis pipeline. You want to be able to generate data that you can give to that analysis pipeline where you know what the answer should be, and then make sure that the analysis pipeline gives you back that answer. With imaging data, that's hard, right? Generating simulated imaging data is pretty challenging still. There are tools, but they're not great. For like behavioral or other types of data, it's often much easier. Another thing you want to do in general, and this is often a little bit easier, is like what we call null or randomization testing. The idea, so let's say that you're trying to look at correlations between some brain feature and some behavioral measure in a large sample. What you could do is take that data set and create a synthetic version of it where basically, like, for example, subjects are just randomly reordered. And then rerun that from the very earliest, like from the rawest raw data, you want to do that, and then run it through your entire pipeline and ask at the end, is it still giving you an answer, right? So for example, if you're doing this, if your pipeline, if you're doing machine learning and you have leakage, you're going to get a good result at the end of that pipeline, even if you randomize the data. That's how you know that there's leakage going on. And so that's another really important tool. And so one of the cool things is that there's now like pretty good tools for generating synthetic data that like look rough. They have like the same types of distributions and same types of features as your actual data. Like this synthetic data vault thing is something we've been using a good bit. That's pretty handy. So it's become a lot easier to make like realistic synthetic data. So I definitely suggest learning more about how to do that. Okay, so just to wrap up, we still have a few minutes for questions. I hope I convinced you that I think many of you probably knew about the reproducibility problems coming into this. If you didn't, I think I've given you an introduction to them. But I should say that it's not really any different in neuroscience and psychology than it is in many other areas that have complex big data and complex workflows. And I think in general, people in kind of AI now are really grappling with the degree to which like a lot of the results in AI might suffer from many of the same reproducibility problems. Statistical power is a clear part of the issue, and shared data is helping us sort of address that to some degree. But I think that with as code becomes increasingly important for everything we do, knowing how to write code in a way that's going to like make sure that you are likely to get the right answer just becomes a really important thing. And so that's why learning about the tools that you're going to learn about here is a really important thing. So thanks. Time for questions.
Yeah, I was wondering, what's your favorite programming language and why?
Okay. For a long time, my favorite programming language has been Python. Not that it's the great, I think, I like it mostly because it has a lot of good tooling. Pretty much anything that I want to do, except for some statistical stuff. Python has a really good ecosystem for machine learning stuff, for neuroimaging stuff. So we do all of our neuroimaging workflows in Python in the lab now. The other thing that it has that for example, MATLAB doesn't have is like really good software engineering tooling. And increasingly, it also has the best ecosystem of like AI tooling. It's not, there are things that I don't love about it, right? It's not the most kind of robust language in terms of like preventing errors. Like there are languages, like people are very excited about Rust right now as a language. It's both faster and like much more protective, but it's also harder to write code. The nice thing about Python is that like it's pretty easy to write code. Another nice thing about Python is that there's so much Python online that the AI tools have a lot of Python in their training data. The one flip side of that, and I'll show an example of tomorrow, is that there's so much bad Python online that they often will end up solving problems in bad ways because they saw a lot of examples on GitHub. But in general, yeah, Python is the language that we choose. We use the one. So what are the exceptions? For statistical stuff, we'll sometimes do stuff in R. And fortunately, it's easy to mix and match, especially with like Jupyter Notebooks. It's easy to mix and match Python and R. That's pretty much the main exception. I mean, we just have in JavaScript for like online experiments. But yeah, those are kind of the choices for us.
Thank you.
Yeah.
I'm curious, in the study with 70 teams where they all had the same data set, you mentioned that 7 or 8 teams who were negatively correlated with the results, and they did something wrong that you said you weren't going to get into.
Oh
Can I
yeah.
ask what they did?
Yeah, basically, so the design was what we call parametric design, where there are a bunch of different trials that vary in like the amount of gain, right? And what you were supposed to do is build like a regression model that models how much does brain activity change as a function of gain. But that needed to be centered, which basically really what they did was they built a regression model without an intercept. And so you could have like, a voxel that was just activated, but didn't change with respect to gain. But because they didn't have an intercept, it thought that that was a gain effect when really it was just an intercept effect. Yeah. So that was basically what they did. So a very simple mistake, right? Okay. Nobody would ever do that in like a regular world regression, but in an fMRI model, it's a little trickier. So yeah.
You made a comment on when collecting data sets, if you have a limited data set, you should focus on what you can answer with your data, not what you want to do with that. Do you have an example where you've come across that? You had to switch what you wanted to do because the question you wanted to answer you couldn't reach with sense.
Yeah, that is interesting because when the comment was really more focused on like at the planning phase, like before you go do something. We certainly have had, so as may be clear here, I've gone on a journey over the last decade, and that's really changed how I think about doing science. We had papers from 20 years ago that were published like correlations with 16 subjects, with like huge like correlations between brain and behavior of 0.8, clearly not plausible correlations. And so we've had to change the way that we do things in the lab. And what's happened is like there were studies, particularly studies that we started in the around the time I was writing that 2017 paper, we were planning new studies, right? Or we had just planned and we're starting new studies, group differences with 40 people, right? And then actually we can only get 25, because there's like patient groups. And so we ended up in this situation where we had research that had been done, that when we were planning the research, we thought that it was a reasonable way to do research. By the time the study was done, it was a study that we never would have planned up front, but we had it was part of a big grant with other collaborators. And so a lot of that stuff had just basically gone into the graveyard because we can't figure out how to publish on it in a way that we're comfortable with. So that's why I'm saying, in general, when I made that comment, I was really thinking about like, up front, if you're planning a study, I wouldn't want to go plan something that know to be futile from the beginning. And the main pivots that I have seen there is really people either focusing on just like asking questions with large shared data sets or moving more towards kind of the computational theoretical angle. I don't know if that's helpful. Yeah.
We should learn for open mind and code and data sharing like OSF and GitHub. So probably also open room for imaging data. So I'm just wondering how you take advantage of those different platforms.
Yeah. So what are the things that we generally use? We generally use for sharing of imaging data, we always use OpenNeuro. Anything that's in the BIDS format, we would share using OpenNeuro. We use OSF for pre-registration primarily. I don't love the OSF interface for like data sharing. Sometimes we will share data. Some of the trainees really want to use OSF to share data. I prefer using Zenodo, which is a data sharing platform run by CERN in Europe. for sharing of data and for sharing of code. So you can share data. I have a whole thing in my book about how to share code and data, but you know, a lot of people just give you a GitHub link, right? A GitHub repository. Problems with that are, you know, one, like you give me a repository, I don't know exactly which version you used in order to, you know, to do the thing that you're publishing. Is code can change. I also don't know if you know, you could delete your GitHub repository and then I wouldn't have any access to it. So Zenodo has this cool thing where if you can log into Zenodo using your GitHub credentials, and then you can basically go link your Zenodo your Zenodo account to your GitHub for, let's say, for this particular repository. And then if you go to GitHub and create a release, which is like a snapshot of your repository, it automatically puts that into Zenodo and creates a DOI that you can put into your paper to cite it. So that's our preferred way of actually sharing data and code. Yeah.
I was wondering, you said, another, solution to the reproducibility is the going computational theoretical. What do you mean by that? Like different trying to which models best describe behavior or best, yeah.
Or building like trying to build computational models to give you more insight into things. So, you know, I had a postdoc who ended up spending most of his time kind of building sort of dynamical neural network models to try to understand, you know, cognitive tasks, ultimately with the goal of linking those up with brain activity. But because we didn't have the money to go collect the brain activity we would need to do it, and he had a large behavioral data set that he could do it on. We basically, you know, did that to try to come up with ways to try to understand how might the task be done that could then be used in imaging analysis down the road. That's one example.
Speaking more about the generalizability of the results, in particular for fMRI, like how would you say that noise is playing an impact on these particular measurement noise and feature variability?
Sorry, what exactly are you asking about the effect of?
Like, how much is noise affecting these?
What kind of, what are you thinking when you say noise?
Noise in the measurement of the signal.
Yeah. Clearly you know, that's the fundamental problem for us. And, you know, so part of what people have been trying to do in, you know, MR acquisition for a long time is figuring out how can we improve the signal to noise ratio of our measurements. One of the big things that has allowed that in recent years is the move to higher field strength, like 7 Tesla imaging, because that does increase the signal to noise ratio, holding everything else constant. Or you can like trade off signal noise for smaller voxels or things like that. You know, the reason I asked what you meant by noise, right, is because that like there's like real, like, you know, there's noise in the kind of like, you know, really randomly distributed stuff. There's also kind of like artifact which is like head motion effects or things like that, which are like, you know, they're signals in the data, right? They're not noise in the sense of just like random measurement error. They're systematic errors in the data, but that I think actually often have a much bigger impact on the way that people analyze data. Certainly in the resting fMRI literature, you know, we've seen that head motion has become like a huge issue in terms of like how to, you know, what are its impacts? What's the best way to deal with it without like killing signals of interest? So I think a lot of us worry less about kind of like the random noise part and more about the kind of systematic error part from, you know, from kind of confounding signals like head motion. Yeah.
One extent, or when, you know, people can do all sorts of processing steps. If you ask them to do a bunch of the data visualization before doing that, to what extent that could normalize what people are doing, or just also getting them to do it more correctly? I'm saying this because recently I've had this experience with this data that comes about pre-processed and everything, but when I was plotting it, there was this very interesting trend that seemed like they didn't detrend the data, and the data had an obvious pattern, which was giving me some very nice results, but I got a little skeptical. So I was wondering if you had, for example, like before people do all these experiments, if you had them some like mandatory visualization steps, do you think that it would be that people would kind of converge to a more correct set of steps.
I think, I mean, depending on what exactly you mean by correct, I certainly agree that, like, in general, people need to look at raw data and at process data more deeply than they have. And I think, one of the kind of like the bad impacts of like a lot of the focus on kind of p-hacking and stuff like that is it's made a lot of people feel like they can't look at their data at all. But I think that looking at the raw data and really making sure that there's nothing funky going on there is really important. So we have a tool that we built called MRI-QC, which is a quality control tool for imaging data, which is meant to kind of help people do this because you have to kind of like you have to know what to look for, right? And so we have that tool. We also, fMRI Prep puts out a bunch of reports, right? fMRI Prep is kind of I don't know how many of you are familiar with fMRI Prep. It's basically a workflow for pre-processing of fMRI, right? It's kind of a black box, right? You put a data set in, you get pre-processed data out. But the goal is to not just have people do that, but to have them look at the reports at each of the steps where things can go wrong. And we try to give visualizations that make it really easy to see if something has gone wrong. So I think that, yeah, any kind of visualization one can use to basically make sure that the data are acting the way that they're supposed to act and make sure that the processing is acted the way that it's supposed to act. I think it's really important. Yeah.
So we're basically at time.
Okay.